Today we're going to look at the basic anatomy of the sheep brain. Let's begin by orienting ourselves. This is the anterior of the brain. This is the posterior. The top is the dorsal surface of the brain, and the bottom is the ventral surface of the brain. As you read in your text, there are 6 major brain regions. From the dorsal view, we can see the cerebrum and the cerebellum. From the ventral view, we see the diencephalon, the midbrain, the pons, and the medulla oblongata. Looking at the brain from the dorsal view, notice the longitudinal fissure which separates the cerebrum into a right and a left cerebral hemisphere. The cerebral hemispheres are further subdivided into units that are called lobes. The lobes are named based on the bones that overlie them. So we have the frontal lobes, the parietal lobes, the occipital lobes, and the temporal lobes. The surface of the cerebrum is folded into gyri, and the gyri are separated from one another by these dark grooves called sulci. Adhering to the surface of the brain is a thin, transparent layer called the pia mater. The pia mater is the innermost lining of the meninges, and it covers the cerebral cortex, or the gray matter of the cerebrum. As we look at the ventral surface of the brain, notice the 2 olfactory bulbs which connect to the cerebrum. Each olfactory bulb leads to an olfactory tract. The optic nerves and the optic tracts merge at an X-shaped structure called the optic chiasm. Near the optic chiasm are the mammillary bodies. This small hole is the infundibular canal. The optic chiasm through the mammillary bodies is the region that we call the diencephalon. These large bumps are the cerebral peduncles, and they are the inferior markers for the midbrain. Together, the midbrain, the pons, and the medulla oblongata are referred to as the brain stem. Looking at the posterior view of the brain, notice the transverse fissure which separates the cerebrum from the cerebellum. If we pull these 2 regions apart, we can see the 4 bumps that are collectively called the corpora quadrigemina. These are the posterior markers for the midbrain. The 2 larger bumps are the superior colliculi. The 2 smaller bumps are the inferior colliculi. And if you look closely, just above the superior colliculi, you can see the pineal gland. From this view, we can also see the surface of the cerebellum. These fine folds on the surface are called folia. From the sagittal view, we can easily see the corpus callosum, the structure that connects and allows for communication between the 2 cerebral hemispheres. Below that is the lateral ventricle. In the lateral ventricle, we have the choroid plexus, this dark brown stringy mass. This region is the diencephalon. The diencephalon is made up of the thalamus, the epithalamus, and the hypothalamus. This round circular structure is the intermediate mass of the thalamus. The indistinct region below the intermediate mass is the hypothalamus, and this small piece of tissue that connects to the pineal gland is the epithalamus. The space around the intermediate mass is called the 3rd ventricle. The 4th ventricle is located between the cerebellum and the pons. The 3rd ventricle is connected to the 4th ventricle by way of the cerebral aqueduct, which passes through the midbrain. From this view, we can also see the arrangement of white matter and gray matter in the cerebellum. The white matter is arranged in this tree-like structure called the arbor vitae, and the gray matter is the darker tissue, which is called the folia. Those are the basic structures of the sheep brain. Please read the dissection directions in your lab manual and be prepared to do the dissection in class next time.